Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF entrance exam question paper conducted in the year 2021. I have divided the question paper into two parts. So this is the first half of the question paper. So coming to the first question. A mutation that generates a termination codon is known as. So it is known as a nonsense mutation. So a nonsense mutation occurs in DNA when sequence change give rise to a stop codon rather than a codon specifying a particular amino acid. And the presence of this stop codon, it results in the production of shortened protein which is likely to be non-functional. Then next one. Which one of the following is an achiral amino acid? Naturally, it is glycine. Glycine is the only achiral amino acid among the amino acid series. So it has got two hydrogen atom as its side chain. So it is A. Then next one. In an enzymatic reaction following Michaelis-Menten kinetics, doubling the substrate concentration from S0 to 2S0 resulted in an increase in the rate of reaction by 2%. Which of the following statement is true for this? Okay. Now this is your Michaelis-Menten graph. So this is concentration of substrate versus concentration of velocity. So when you doubled the substrate, this is the Km value, this one. Km value. So when you doubled the substrate concentration, what happened? Well, the reaction rate increased only by 2%, not more than that. So we can say that even as the substrate concentration is doubled, there is only a negligible increase in the velocity value. That means it is very near the Vmax value, that is over here, because it is stabilized when it reaches the Vmax value. So it must be somewhere around here, or it has reached the saturation level. So a system is saturated only when the concentration of substrate is well above the Km value. So over here only the system is saturated, and this is your Km value. So it is well above the Km value. So even S0 by 2, half of the substrate concentration will be more than Km. So in all 1, 2 and 3, they have given a Km as more than substrate concentration. So that is not correct. So our answer is option 4. Km is very much less than S0 by 2. Answer is option. Then next one. You are preparing 100 ml of a solution containing 100 millimolar Tris-HCl, 5 millimolar MgCl2, 1 millimolar DTT. If the stock solution provided are 1 molar Tris-HCl, 100 millimolar MgCl2, 50 millimolar DTT, the amount of each component would be. So the answer here is option 2. I have done the calculation in the next. So these are the stock concentration, and these are the working concentration. Now Tris, you need 1 molar. From that, how much you have to take? This is your stock. So M1V1 is equal to M2V2. So from 1 molar Tris-HCl, how much you have to take to get 100 millimolar Tris, or 100 into 10 raised to minus 3 molar Tris? That will be 10 ml. Similarly for MgCl2 and DTT. And you will get the answer 1 molar Tris-HCl 10 ml. 100 millimolar MgCl2 5 ml and 50 millimolar DTT 2 ml. Next one. For sequencing DNA by Sanger's method, the chain elongation is terminated by. So DDNTPs or dideoxynucleotides are chain elongation inhibitors of DNA polymerase used in the Sanger method of DNA sequencing. And they are also known as 2', 3' dideoxynucleotides, because both the 2' and 3' position on the ribose lack hydroxyl group. So the answer here is 2', 3' dideoxynucleotides. Answer is option 2. The next one is match the following. Methylation of lysine. Methylation of lysine activates gene by modifying histones in chromatin. Lysine 9 is modified in histone by methylation. Then hydroxylation of proline, that is collagen structure. Phosphorylation of tyrosine will cause cell signaling, and polyubiquitination of lysine will target a protein for degradation. So the answer here is option A2B1, A2B1, C4 and D3. So the answer is option 3. Then next one is also a match the following components of list 1 with those of list 2 under physiological condition. So leucine is non-polar, lysine is positively charged, glutamic acid is negatively charged, and glutamine is uncharged polar. So the answer here is option A2, B4, C1 and D3. Answer is option 4. Then, which of the following statement is correct about the reaction catalyzed by serine hydrolases? So, serine protease, that is an example of serine hydrolase, and it has got a catalytic triad, which is histidine, serine, and aspartic acid. And each amino acid in the triad perform a specific task in this process. So here the serine over here, serine as an OH group that is able to act as a nucleophile attacking the carbonyl carbon of the sessile peptide bond of the substrate. So serine is attacking the substrate. A pair of electrons on the histidine nitrogen has the ability, that is over here, over the histidine nitrogen has the ability to accept the hydrogen from the serine OH group. This one. This nitrogen has got the capacity to accept hydrogen from the serine OH group, thus coordinating the attack of the peptide bond. So histidine helps serine in attacking the peptide bond. Then the carboxyl group on the aspartic acid. This is your aspartic acid. This is the carboxyl group. In turn, hydrogen bonds with histidine, making the nitrogen atom mentioned much more electronegative, making this nitrogen atom much more electronegative. So after this you will get what is this is how the reaction is taking place. So an aspartate residue abstracts a proton from serine. No, the aspartate acid residue, it abstracts this hydrogen bonds with histidine over here. This is what is happening. So a nucleophilic serine residue attacks the carboxyl carbon of aspartic acid. Now it is involved in attacking to the substrate. Then an aspartate residue abstracts a proton from histidine. Aspartate, yes, residue abstracts a proton from histidine. This reaction. So 3 is correct. An aspartate residue attacks the carboxyl carbon of the ester to be hydrolyzed. That is also not correct. So the answer is option 3. Then next one. 100 ml of 0.1 normal HCl solution was diluted with 50 ml of 0.05 normal HCl solution. The normality of the final solution is, so N1 V1 plus N2 V2 is equal to N3 V3. So V3 we have as 150 ml, that is 100 plus 50 ml. N1 is 0.1 normal, V1 is equal to 100 ml, N2 is 0.05 normal, and V2 is 50 ml. So substitute in the formula and you will get 0.083 normal. Answer is option 3. Then match the following. Isomerization. Isomerization is rearrangement of atoms to form isomers. Redox reaction. Redox reaction is electron transfer from one species to another. Group transfer. Transfer of a functional group from one molecule to another. Hydrolysis. Cleavage of bonds by addition of water. So the answer here is option D. Then which one of the following cannot be used for differential gene expression analysis? So what is differential gene expression analysis? It is a biochemical process that determines which genes are actively transcribed and translated into mRNA and proteins in a cell and under what conditions. So under what conditions a particular gene is being transcribed, that is mRNA is produced, and translated, that is protein is produced. That is called as differential gene expression. So they have asked which one of the following cannot be used for differential gene expression analysis. So it is whole genome sequencing data analysis. That itself is the answer. EST data is express sequence tags that can be used. Microarray, yes, and mRNA sequencing data analysis, that also can be used for differential expression analysis. Then next one. If the codon for the translation of mRNAs to proteins were 4 letters long instead of 3, what would be the maximum number of hypothetical amino acids that could uniquely be recognized by the tRNAs, assuming only one codon as stop codon? So here they have asked, instead of 3 codons, if it was a 4-letter codon, then how many hypothetical amino acids could be recognized by tRNAs, and you have only one stop codon. So if it was 3, we know that it is 4 cube, that is 64. So similarly, if it was 4, it will be 4 raised to 4, that is 256. So 256, out of that, 1 is termination codon. So tRNA doesn't carry any amino acid for that. So it is 256 minus 1, that is 255. Answer is option 2. Then next one. Beta-lactam ring is present in. It is present in penicillin. Answer is option 1. Which one of the following amino acid has a strong tendency to disrupt alpha helices and beta strands? So it is naturally proline. Answer is option 4. Then which one of the following is not a characteristic of collagen fiber? It is the main component of skin, bones, tendons and teeth. Yes. It is a fibrous protein with alpha helical coiled coil structure. Yes. The core of the triple-stranded helix is populated with glycine residue. That is also correct. So what is left out? Each of the strand is independently stable due to the presence of hydrogen bonds. That is not correct, because intrachain hydrogen bonding is not possible in collagen. It is 3 left-handed helical strands twist to form a right-handed triple helix. That is what is called as a collagen fiber. So in that 3 left-handed helical strands twist to form a right-handed triple helix. So answer is option 4. Then next one. Match the components of list 1 with those in list 2. Neutrophils. Neutrophils are phagocytose and destroy bacteria. Platelets. Platelets initiate blood clotting. B cell make antibodies and T cell kill virus infected cells. This was a very easy one. So A is 2, B is 4, C is 1 and D is 3. Answer is option 4. Coming to the next question. Breeding capital A small a and capital A genotype resulted in 5 offsprings. What is the probability that exactly 4 offspring will have the genotype capital A? So AA into AA you will get on crossing, half of them will be of AA genotype. Now you have to get out of exactly 4 offsprings out of 5. So we have to see the possibility. Suppose we take capital A as A and capital A small a as H. Then these are the 5 possibilities that are possible. So what is the probability that exactly 4 offsprings will be of A genotype? Will be half into half into half into half into 5. So 5 by 32. So the answer here will be option 4. Then next one. Which one of the following molecule causes catabolite repression of lac operon? It is glucose. Answer is option 3. Which nuclease of bacteria is responsible for degrading the genome of the invading lambda phage? So it is restriction endonuclease. Answer is option 3. DNA fingerprinting for forensic investigation is based on. So it is based on variable number of tandem repeats and these are repetitive sequences. Answer is option 3. Then at which phase of the growth curve are bacteria more sensitive to ampicillin? So that is during the log phase they are more sensitive to antibiotics. So answer is option 2. Then next one. You are performing a PCR reaction in which you need to use 20 picomoles of each primer. If both the primers are 20 nucleotides long and the average molecular weight of each nucleotide is 300 Dalton, the amount of each primer required for 100 microliter reaction. So you are performing a PCR reaction and you need 20 picomoles of each primer, that is forward and reverse primer. And they have told that both the primers are 20 nucleotide long and the molecular weight of each nucleotide is 300 Dalton. Then the amount of each primer required for 100 microliter of a PCR reaction. So I have given it in the next slide. So each primer is 20 nucleotide long and molecular weight is 300 Daltons. So one nucleotide is 300 Dalton, 20 nucleotide will be 6,000 Daltons. And we know that 1 Dalton is 1 gram per mole. So 6,000 Dalton is 6,000 gram per mole. That means 1 mole has 6,000 grams. So 20 into 10 raised to minus 12 moles, they have given as picomoles, will have 6000 into 20 into 10 raised to minus 12 grams. So that will be 12 into 10 raised to minus 8 grams in 1 ml. So we have to see for 100 microliter reaction or 0.1 ml. So 12 into 10 raised to minus 8 into 0.1, that is 12 into 10 raised to minus 9 grams. Now convert to nanogram. 1 gram is 10 raised to 9 nanogram. So 12 into 10 raised to minus 9 gram will be 12 nanogram. Answer key, it is 120 nanogram, but I am getting the answer as 12 nanogram. This is how the calculation is done. But I don't know whether I have made any mistake in my calculation. If you find any mistake, please let me know in the comment section. Okay, next one. The blood volume of an individual is 5 liters. The person was injected with 500 mg of a drug that has a molecular weight of 100 Dalton. If the drug is metabolized so that half of the drug remains in the bloodstream after every passing hour, the concentration of the drug of 4 hours after injection is. So after each hour, they have told that the drug is metabolized so that half of the drug remains in the bloodstream after each hour. So initially it was 500 milligrams. Then after 1 hour it will be 500 by 2, that is 250 mg. After 2 hours it will be 12.5. After 3 hours it will be 62.5 and after 4 hours it will be 31.25 mg. So weight, we got it as 31.25 mg. Now we have to see for the concentration. They have given the molecular weight as 100. Now we have to see for molarity. That is WB by MB into V. So 31.25 into 10 raised to minus 3 grams divided by 100 into 5. You will get it as 0.0625 millimolar. So the answer here is option 4. The next one, in an electron transport chain, which one of the following can be a 2-electron carrier? So out of these, it is flavin, because if you see, you will your electron transport chain, you can see that FAD plus 2 electrons plus 2H plus will give FADH2. So flavin is the 2-electron carrier. Then next one. The E. coli cell has a volume of 1 micrometer cube and the volume of single base pair is 1 nanometer cube. If the E. coli DNA has 5 into 10 raised to 6 base pairs, then the volume occupied by the genome in the cell is. So volume of one base pair is 1 nanometer cube or 10 raised to minus 9 meter cube. And DNA is 5 into 10 raised to 6 base pair. So volume occupied by 5 into 10 raised to 6 base pair is 5 into 10 raised to 6 into 10 raised to minus 9. That will be around 5 into 10 raised to minus 3 meter cube, that is 0.5%. Answer is option 2. Then next one. Which one among the following can be effectively transmitted from person to person due to coughing? So swine flu and adenovirus. Yes, that can be easily effectively transmitted, whereas AIDS cannot be transmitted. Dengue is through and malaria is dengue and malaria by mosquitoes. So it is not through coughing. So answer is option 1, that is swine flu and adenovirus. Then during replication, DNA polymerase. So DNA polymerase can initiate DNA synthesis de novo. No, it cannot synthesize de novo. It requires a primer to initiate DNA synthesis. So cannot initiate DNA synthesis de novo. That is the answer. Option 2 is the answer. Then next one, conversion of L-pyruvate to L-lactate is an example of reduction. Conversion of UDP galactose to UDP glucose occurs by oxidation and reduction. Then next one, a 2 kb insert has to be ligated to a 8 kb plasmid in a ligation mix where we want to keep the vector insert molar ratio as 1:2. If 1 microgram of vector is used, the amount of insert to be used, so nanogram of insert, will be equal to this nanogram of vector into kilobase pair size of the insert by kilobase pair size of vector into molar amount of insert by molar amount of vector. So you have to convert this 1 microgram vector to nanogram. So 1 microgram is 1000 nanogram. Now substitute in the formula and you will get it as 500 nanogram or 0.5 microgram. Answer is option 3. Then next one. During translation, which one of the following amino acyl tRNAs binds first to the P site of bacterial ribosome? So it is F-met tRNA F-met. It is the methionine carrying tRNA that binds to P site. F-met is formilated methionine and is specifically used for initiation of protein synthesis in bacteria. Now coming to section B, that was about section A, and in section A I have not answered physics, chemistry and mathematics numerical questions. Now coming to section, I have answered only the biology questions. Okay, now coming to section B. DNA replication requires DNA topoisomerase to remove the supercoiling of DNA that accumulates at the end of a growing replication fork. You wish to perform a PCR amplification of a gene that has been provided to you in a 6 kb plasmid vector. Why will you not use topoisomerase in your PCR reaction mix? So they are conducting it usually in normal condition that you need DNA topoisomerase to remove the supercoilings when DNA is undergoing replication. But you wish to perform PCR amplification of a gene that has been provided to you in a 6 kb plasmid vector. So your gene is inside a 6 kb plasmid vector and you will not use topoisomerase in your PCR reaction mix. Why? So the question is, why will you not use topoisomerase in your PCR reaction mix? That is the real question. The other things are just an explanation or just an addition to the question. Okay, now why will you not use topoisomerase? Because PCR is in vitro condition. The DNA has already been isolated from the cell. So PCR is conducted under in vitro condition and the answer is denaturation step in the PCR protocol precludes formation of supercoils. Answer is option 2. Then enzyme linked immunospot, ELISpot, assay measures. So enzyme linked immunospot assay is a sensitive method for quantification of the number of cytokine secreting cells. So the answer here is number of cytokine releasing cells at single cell level. Then next one. Which one of the immunoglobulin causes type 1 hypersensitive immune reaction? So type 1 hypersensitive is also known as immediate reaction and involves IgE mediated release of antibodies against the antigen. The result is mast cell degranulation, release of histamine and other inflammatory mediators. So which one of the immunoglobulin causes type 1 hypersensitive immune reaction? It is IgE, option 1. Then you have recently observed a mutation in gene X in patients with lung cancer. You have sequenced the gene X in these patients and has observed that both the copies of gene X are mutated and the mutation leads to absence of protein X in the patients. The gene is most likely. So here you have observed a mutation in gene X in patients with lung cancer. So what you did, you isolated the gene and sequenced it, and you observed that both the copies of the gene X are mutated and the mutation led to the absence of protein X in these patients. So which gene it is most likely to be? Oncogene. So an oncogene, it is a mutated gene by itself and has the potential to cause cancer. Then tumor suppressor gene. These are normal genes that slow down cell division, repair DNA mistakes, or tell the cell when to undergo apoptosis. So when a tumor suppressor gene doesn't work properly, cells grow out of control, which can lead to cancer. So naturally, the mutation might be to the tumor suppressor gene. So answer is option 2. Then next one. Which one of the following strategies do viruses employ to evade the human immune system? So virus infected cells show reduced expression on the surface MHC class 1 molecules. That is the answer. And next one. A protein cargo X is destined for lysosomal degradation in cells under specific condition. This can be tracked by red fluorescence emitted by the tag when it reaches lysosome. So when the protein cargo X reaches lysosome, it will be in contact with the acid phosphatases as well as the acidic pH condition inside the lysosome. And this lysosomal degradation, it can be tracked by red fluorescence that is emitted by the tag which is attached to the protein cargo X. Okay. Now what will happen when you treat the cells with NH4Cl? So what is the consequence when you treat with NH4Cl? Now NH4Cl, it makes the environment basic. So acidity degradation by lysosome will not take place, and so no red fluorescence will be emitted. So the answer here is option 2. No red fluorescence will be emitted. Then induced pluripotent stem cells. So induced pluripotent stem cells are type of pluripotent stem cells derived from adult somatic cells, and they have been reprogrammed through inducing genes and factors to be pluripotent. So answer is first one itself, option 1, that is somatic cells are transduced with viral vectors coding for transcription factors that induce a pluripotent state in the recipient cells. The next one. A protein X is active when phosphorylated on tyrosine residues. You wish to mimic this phosphorylation by mutating tyrosine to another amino acid residue. Now, which one of these residues will you mutate tyrosine to? So, this is about what is called as phosphomimetics. And phosphomimetics are amino acid substitution that mimic a phosphorylated protein thereby activating or even deactivating the protein. Within cells, proteins are commonly modified at serine, threonine or tyrosine amino acid by adding a phosphate group. Some non-phosphorylated amino acids appear chemically similar to phosphorylated amino acid. That is, glutamic acid is phosphomimetic of threonine and phosphorylated threonine and glutamic acid have both negative charges. So here the protein X is activated when phosphorylated on threonine residue. Now there is a phosphomimetic for this particular threonine residue, and what is it they have asked. So the phosphomimetic is nothing but glutamic acid is a phosphomimetic of phosphorylated threonine. And threonine and this phosphorylated threonine and glutamic acid, they both have negative charge. So the answer here is option 3. Then next one. You have isolated a hypothetical protein X. X is run on a gel filtration column. The apparent weight of the protein is 80 kDa. When X is run on an SDS page with 2 mercaptoethanol present in the loading buffer, the size is around 40 kDa. So when you passed your protein of interest through a gel filtration column, you got the size as 80 kDa. But when you ran an SDS page under reducing condition, that is by adding 2 mercaptoethanol to the loading buffer, you got a size of around 40 kDa. Now why did you get like that? So mercaptoethanol, it breaks disulfide or reduces the disulfide bonds. So X protein on gel filtration is 80 kDa, and on reduction with mercaptoethanol, the disulfide bonds are broken and it ran as a single band on an SDS page. So it ran as a single band of same molecular weight, that is 40 kDa. So naturally it is a homodimer. Both the bands are of equal size, that is 40 kDa on an SDS page. So the answer here is option 4, that X is a dimer of 2 units held together by disulfide bonds with a molecular weight of 40 kDa per monomeric unit. Then next one. In the following table, list 1 has nucleic acids and list 2 has certain base modification. Match the components from list 1 with those in list 2. So DNA. DNA is 5-methyl cytosine, tRNA is pseudouridine, and mRNA is 7-methyl guanosine. So answer is option 4, that is A2B3C1. Then next one. If a single-stranded DNA sequence of 250 nucleotide consists of 30% thymine, the number of adenine, guanine, thymine and cytosine nucleotide present in it are. So this is a single-stranded DNA sequence. So the number of thymine is may or may not be equal to number of adenine, and number of guanine may or may not be equal to number of cytosine. And Chargaff rules, it doesn't apply to single-stranded DNA. So you cannot calculate the number of residues of adenine, guanine and thymine and cytosine. So answer is option 4, cannot be calculated. Next one. Pyruvate decarboxylase catalyzes the decarboxylation of pyruvic acid to acetaldehyde and carbon dioxide. Its action depends on cofactors TPP, that is thymine pyrophosphate, and magnesium. In this process, the role of TPP in the initial step is to act as. So it is to act as carbanion. Answer is option 2. Then next one. Baeyer-Villiger monooxygenases are flavin-dependent enzymes that catalyze oxidations. Which one of the following oxidation is not carried out by BVMOs? So Baeyer-Villiger monooxygenases are flavin-dependent enzyme that catalyzes the oxidation of ketones and cyclic ketones to esters and lactones respectively. by using molecular oxygen and NADPH. Then BVMOs also catalyze sulfoxidation and N-oxidation. So out of these, the answer is option 4, that is aldehydes to carboxylic acid. That is not carried out by BVMOs. Then next one. Which one of the following is a malnutrition disease? It is marasmus. Marasmus is due to the deficiency in calories and energy. Then next one. The fragment obtained from a Sanger sequencing experiment are as follows. These are the fragments obtained. Please identify the template sequence from the above given data. So the sequence obtained after Sanger sequencing is complement of the original template strand. So find for this last one 5 prime 3 prime. Okay. This last one 5 prime 3 prime sequence, you find the complementary of that in the 3 prime 5 prime direction. So it is 3 prime CTT AA TAG TG. That will be your template strand. Answer is option 4. Then which one of the following is not true for local alignment of protein sequences? It is generally used for analyzing distantly related sequences. Yes, it looks for regions or blocks of high similarity between the two sequences. Yes. Gap penalty is not used for insertion and deletion. Actually, it is used. So, which is not true of local alignment of protein sequences is option 3. Smith and Waterman algorithm is used for local alignment of two sequences. That is also correct. Then next one. Following is the table in which list 1 contain names of various steps of gene expression and in list 2 are the enzyme associated with those. Match the components of list 1 with those in list 2. Epigenetic regulation. Epigenetic regulation is RNA polymerase. DNA repair ELF2 kinase. Transcription histone methyl transferase. Translation endonuclease. So the answer is option 4. A4, B3, C2 and D1. Then what is the pH of a mixture of 0.042 molar NaH2PO4 and 0.42 molar Na2HPO4? So pH, they have given the pKa value also. So you can use Henderson-Hasselbalch equation. pH is equal to pKa plus log concentration of the base by acid. And you will get it as 7.86. Answer is option 3. Then next one. Cellulose is a polymer of glucose which is made of dash glycosidic bond. It is beta 1 for linkage. I think it is option 2. They have not given beta over here. Okay. Then which one of the following defines the psi angle in protein backbone? So it is N C alpha C N. Answer is option 1. Then, which one of the following is used to validate the secondary structure of proteins? So, it is the Ramachandran plot. Answer is option 2. Genes that are regulated through gene duplication events are. So, paralogs are homologous genes that have evolved by duplication and code for proteins with similar but not identical functions. So, answer here is option 4, that is paralogs. Then next one. A genetist interested in immune function induces random mutations in a number of specific genes in mice, then determine which of the resultant mutant mice have impaired immune function. This approach is an example of. So you have got what is called as forward genetics and reverse genetics. In forward genetics, you know the phenotype, you discover the gene underlying the phenotype. In reverse genetics, you know the gene and you determine the phenotype. So here what he has done, he has induced mutations to a specific number of genes in mice. So he knows about the genes. and then he is determining what is the resultant mutant mice have resulting mutant mice have impaired immune function. Which of the resulting mutant mice have impaired immune function? And he is determining the function, or he is determining the phenotype of this particular known gene. So it is an example of reverse genetics. Answer is option 2. Then a scientist chose nickel NTA affinity chromatography to purify a recombinant protein. Which one of the following tags was present in his recombinant protein? So nickel NTA columns are used for purification of recombinant proteins with polyhistidine tag on either terminus, that is either N or C terminus. And the most common polyhistidine tag is hexahistidine tag. So answer here is option 4, hexahistidine. A recombinant protein with the hexahistidine tag has high affinity for nickel, whereas other proteins will either bind with low affinity or they will not bind at all. Then you do a washing for that particular column and with the different concentrations of the buffer, and you can elute out the recombinant protein and also the proteins either not bound to the column or bound with low affinity to the column. So answer here is hexahistidine. Then during eukaryotic replication, dash degrades the RNA primer by 5' 3' exonuclease activity. It is FEN1. Then next one. The presence of cardiolipin is characteristic of the membrane of mitochondria. Answer is option 4. Then the enzyme used to prevent unwanted self-ligation of DNA molecules during cloning experiments is. The vector DNA should be dephosphorylated to prevent self-ligation. And dephosphorylation, it is commonly carried out by using the enzyme alkaline phosphatase that remove the phosphate groups from the 5' ends of the DNA. So the answer here is alkaline phosphatase. In genomics, a contig means. So a contig means a set of overlapping fragments that form a continuous stretch of DNA. Answer is option 1. The hypochromic effect is used to estimate the melting temperature for double-stranded DNA. It arises because. So the purine and pyrimidine bases in DNA strongly absorb UV light. Double-stranded DNA absorb less strongly than denatured DNA due to stacking interaction between the bases. So they have asked the hypochromic effect is used to estimate the melting temperature for double-stranded DNA. It arises because of stacked bases in double-stranded DNA absorb less UV light than unstacked bases in single-stranded DNA. Answer is option 2. So double-stranded DNA is having stacked bases, whereas single-stranded DNA, it doesn't have base stacking. Gel filtration chromatography separates protein on the basis of. So it separates so size exclusion chromatography, it is a method where separation of different compounds occurs according to their size. That is hydrodynamic volume measured by how efficiently they penetrate the pores of a stationary phase. So the answer here is option 2, that is hydrodynamic volume. Then next one. Fake medicines are a nuisance that shatter the faith of patients in medicine and enhance the emergence of drug resistance. A medicine is likely to be fake if. Answer is option 2, that is same peak intensity and same retention time are not observed on injection of equal amount of the test and standard sample on HPLC. Then next one. Chip on chip, a technique that combines chromatin immune precipitation with microarrays is used to identify. So chip on chip assay is used to investigate interaction between protein and DNA under in vivo conditions. So out of these, transcription factor binding regions in the promoters is the answer. That is an example of DNA, protein DNA interaction. The next one. You have an assay method that can estimate a compound A up to level 10 mg per ml. If you need to modify it so that you can estimate 0.1 mg per ml, you need to improve upon. So what is sensitivity and specificity? Now sensitivity, it refers to the probability of a positive test conditioned on truly being positive, and specificity refers to the probability of a negative test conditioned on truly being negative. So in a diagnostic test, sensitivity is a measure of how well a test can identify true positives, and specificity is a measure of how well a test can identify true negatives. So higher sensitivities will mean lower specificities and vice versa. So here, in order to improve, you need to in order to modify, you need to improve upon the sensitivity. So answer is option 2. Then next one. Which of the following is the most effective reducing agent of disulfide bonds in protein? So it is DTT, dithiotretol. Answer is option 1. Because it is stronger reducing agent than mercaptoethanol, and in addition, mercaptoethanol will be oxidized easily upon exposure to air, whereas DTT will not. Then next one. Digitalis is used for the treatment of congestive heart failure because it can increase the force of contraction of heart muscles. Answer is option 3. Then next one. A 20-mer peptide composed of 20 coded standard amino acids was hydrolyzed with 6 normal HCl. So this is a hypothetical peptide, and it was hydrolyzed by 6 normal HCl. However, only 17 amino acids were detected when the hydrolyzed was analyzed by chromatography. Since you have used a 20-mer peptide, you need to and coded by 20 amino acids, you should have got 20 amino acids when you analyzed it by chromatography after the peptide was hydrolyzed with 6 normal HCl. So when you treat a peptide with 6 normal HCl, this is how you will get that amino acid. The peptide bond is broken and you get distinct amino acids. This is how you should have got. So when you use 20 mer peptide, you should have got 20 amino acids, but you never got, and 3 amino acids were missing. So which are these missing amino acids? So acid hydrolysis with 6 normal HCl results in addition of water to each covalent peptide bond, yielding the desired individual amino acid. Not all amino acids are completely recovered under hydrolysis by HCl. Some amino acids are hydrolyzed to their acid form, that is asparagine and glutamine to aspartic acid and glutamic acid, and respectively. Other amino acids cannot be reliably measured. For example, tryptophan is destroyed during the reaction. So the 3 amino acids are glutamine, asparagine and tryptophan. Answer is option 3. Glutamine and asparagine, they got converted to glutamic acid, aspartic acid, and tryptophan, it was destroyed in the reaction. So the answer is option 3. Then peroxisomes are different from mitochondria and chloroplast mainly because they are surrounded by a single membrane. Okay, answer is option 3. Then copy number variation signifies. So copy number variation, it is defined as a copy number change involving a DNA segment that is 1 kb or larger. So the answer here is option 3, that is DNA segment greater than 1 kb repeated multiple times in the genome. Next one. Which one of the following is not characteristic of an enhancer? So its function is independent of its location in the genome. Yes. Its function is independent of its orientation in the genome. Yes. It is transcribed to form enhancer RNA. Yes. It is conserved in evolution. That is not characteristic of enhancer. Answer is option 4. The enzyme that plays a key role in glucose homeostasis. It is glucokinase. What does it do? So glucokinase function as glucose sensor in the beta cells of Langerhans by controlling the rate of entry of glucose into the glycolytic pathway and its subsequent metabolism. In the liver, glucokinase play a key role in the ability to store glucose as glycogen, particularly in postprandial state. So the enzyme that play a key role in glucose homeostasis is glucokinase. Then next one. Prokaryotic ribosome bind to which one of the following? So prokaryotic ribosome, they bind to Shine-Dalgarno sequence and it is found in bacterial mRNA is a ribosomal binding site. Answer is option 2. Then next one. Which one of the following components of an enveloped virus particle is not encoded by the viral genome? So lipids of the viral membrane are derived from the host cell. So the answer is option 2, envelope lipids. A student clones a gene of interest with the ampicillin resistant genes of PBR322 vector for transformant selection. The student will use. So the PBR322, it is having two different antibiotic resistant genes, that is ampicillin as well as tetracycline. Now what has the student done? He has cloned a gene of interest within the ampicillin resistance gene. So he cannot use or she cannot use ampicillin media for selection of recombinants, because the recombinant will naturally be sensitive to ampicillin. But he or she can use tetracycline plates, because those with ampicillin resistance, ampicillin sensitivity also will grow on, or the recombinants will grow on tetracycline plates. Answer is option 2.